Okay, so I am starting my presentation now. Okay, is my screen visible?
Yes.
So far, so good. This is what we were talking about when cannot reach me. So this is what we were talking about when the last time we concluded our lecture. We ended our discussion on what is called as network of hydrogen bonds. Okay, network of hydrogen bonds can be considered as intricate network of the hydrogen bonds between the protein and the ligand, or the hydrogen bonds that are mediated by water molecule as well. So collectively we can call it as network of hydrogen bonds. Okay, so now let us move on to the next, or you can say next level of our structural analysis of a given protein by uh, by performing surface analysis of protein. Okay, so far so good. I have told you how you can identify hydrogen bonds or different type of interaction in your protein molecule, and what is their contribution in stability of protein as such. Okay, but now onwards we will shift our focus on what are the advantages of visualizing the protein in surface representation. Okay, so what else can this surface visualization of any protein will tell us about the functional properties of that protein as such. Okay, so what are the things that would be enabled for us to learn about the protein if we know its surface? So that would be the major aspect of today's discussion. Okay, तो क्या करने वाले हैं आज अपन? We would see what exactly or what different and interesting things that we can do with protein structure if we are able to visualize its surface. Okay, can we map some interesting properties of protein onto the surface that will let us understand how the protein might function or how it might influence other molecules in its environment as such. Okay, so first I would start out with a demo by which I will show you one of the way how do we calculate surfaces of protein. Okay, so we would be using our favorite molecule 1-HEW, which is lysozyme from egg white of hen. Okay, so so far so good. We have dealt with our molecule in CPK representation, right? अपन ने अब तक जो कुछ भी एनालिसिस किया सब सीपी के रिप्रेजेंटेशन ने किया, but there is also it is also possible to visualize the surface of your protein as shown in the image on its vicinity. उसके बाजू में जो इमेज है, the same protein as you have visualized in CPK, if you visualize the same protein in surface, it would look something like this. Okay, this panel that I am highlighting now. So by visualizing the surface, of course you can detect cavities in your protein. Okay, you can detect you can figure out ki, okay, what might be the binding pockets that might be present on my protein by detecting the surface of the given protein as such. Okay, and you can have surface representation with various with with various levels of resolution. Okay, so over here in this middle figure you can say that I have rendered the surface of this protein with surface quality of 1. Okay, on the other hand, this particular image, the third image on the slide, I have rendered the surface of this protein with surface quality 2. So now, as I increase the resolution of the surface, you can detect more details of the surface in this rendering while surface quality 2, while very less details of the pockets and cavities are visible when you try to detect the surface of the protein with surface quality 1. Okay, so first I will show you how these things are detected, or you can say how these things are done. Okay, now I will be shifting our software from spdbv to another software called as pymol, because pymol is way better for representing the surface of a given protein as compared to your spdbb. Okay, so let us begin our discussion with identifying the pymol asset. Okay, so here we go. Okay, now I will start our pymol, or maybe I will just open it in pymol. My default program is pymol itself, therefore if I double click it, I will be seen with the molecule. Okay, now can everyone see this?
Yes, sir.
Okay, so in this representation, it is formal ball and stick representation in CPK model. Okay, the only difference over here is that we have colored the, by default, pymol colors the protein carbon atom with green color. On the other hand, in CPK representation, if I color it by elements, oh, I don't see it here. Uh, so this is what you must be familiar with. Okay, so this is the CPK representation that you see in SPDBV. Okay, okay. Well, now in order to visualize it for surfaces as such, what you can do is that you can hide all the lines that has been shown. Okay, you can hide all waters as well, and then you can show it as surface. Okay, but still you don't see that one HEW molecule. Okay, so well, let us visualize, or what you can do is that you can color it in the shade of white. Okay, so now it is the same molecule as you can see over here, but you somehow are not able to visualize your trinag inhibitor. तो उसके लिए अपन क्या करते हैं, what we usually do is that we go to this sequence and select the trinag molecule. Okay, and maybe we will create an object out of it so that we can do anything that we wish with it. Okay, so now this object is nothing but your trinag. We don't require this sequence now, उसको जाने देते. We would again zoom on the protein molecule, and now I will show this object, which is our inhibitor, in sticks representation. Okay, and I can color it by like this. I will color all the carbon by yellow, and it is still in your stick representation. That is how you see this, right? So you might have seen such beautiful images in your biochemistry textbook, right? लेनिंजर में या वोल्ट एंड वोल्ट में आपने ऐसे इमेजेस देखे होंगे, right? So now this is the surface representation at surface quality 1, which is by default shown in your PyMol. Okay, now let me change the surface representation to surface quality 2, right? I can give the command in PyMol over here. Okay, what I'm doing over here is that I'm setting the surface quality of the protein with 2 at the second level of resolution. So if I hit enter, you know, you will visualize a rendering bar over here. Okay, so it says that the computation of rendering at the second level of surface quality is in progress, and now you can see you can identify the inhibitor binding pocket quite visually over here. इसके अलावा you can also detect if there are certain cavities which are present over here. Okay, so you can render the surface of your protein as per your requirement. At what level do you want to have details of these surfaces? Okay, so this is how you represent your molecule in surface representation. Okay, yes, of course, this PyMol is also a freeware. Okay, like SPDBV, you can download it on Delano Scientific Network's website. Okay, it is an open access software, so you can use it for free, I suppose. Okay, so there would be no issue for you while working on this thing. Okay, so everyone now got how do we represent the surface representation of your protein?
Yes, sir. Yes.
Okay, okay, good. So let us move on. So I have shown you the demo for calculating the surface for this one HEW molecule, and over there you have seen how this particular inhibitor occupies the surface of your protein. Okay, by default, if you use this line model or ball and stick model, somehow you won't be able to identify the pockets and everything, which are one of the major feature of protein ligand interaction. Okay, so well, let us move further. So what now? I know how do we render the surface. So what? तो क्या होगा सर, मुझे अगर ये पता चल गया है कि सरफेस कैसे बनानी है. So what you usually do after you know how to map the surface of protein, what you can now do is that you can map some interesting properties on the surface of your proteins. Okay, and some of the properties that can be mapped onto the surface of the surface of your protein are in form of molecular hydrophobic potential, what we call it as MHP. You can calculate and map the electrostatic potential on the surface of the protein, as well as you can also detect the binding site for the given proteins. Okay, now how do we detect the binding site? Usually the binding site are the deepest cleft in your proteins. Okay, so by enabling the visualization of your protein in surface representation, you can actually visualize how many pockets are there in your protein that can actually go and bind to certain inhibitor, or say certain substrate, or say certain prosthetic group, or even cofactor maybe, right? So it can be used to predict what might be the binding site for different non-protein molecules on your protein surface as such. Okay, so the major focus would be on discussing what exactly is molecular hydrophobic potential and what is its significance. Okay, how can we map this molecular hydrophobic potential on the surface of protein, and what is to be inferred by such an analysis. So that is the major uh, major thing that we are going to do today. And I will, of course, introduce with the concept of what is electrostatic potential and what can we infer by mapping this electrostatic potential onto the surface of the protein. Okay, I think someone has asked to join. Okay, welcome Chandekar madam. Okay, so let us begin with mapping this surface property. Let us begin with discussing what is molecular hydrophobic potential, or MHP in short. Okay, so now this molecular hydrophobic potential, or what we call it as MHP, is calculated to describe hydrophobic effects in biomolecule. Okay, so as I have already told you, that when your ligand goes and attaches to a given protein, it might stabilize this protein ligand complex by forming different hydrogen bonds, by forming, maybe if it is charge, it can form charge ionic interaction with the protein molecule. Okay, if the prosthetic group is supposed to be working in close association with the protein molecule, it can also form the covalent bond with the protein molecule. Well, say for example, in case of PLP dependent enzymes, the PLP is usually attached covalently to the protein molecule with, say, some lysine residues preferably. Okay, that means this PLP covalently attaches to the protein. Okay, or such protein prosthetic group or ligand group complexes can be stabilized using hydrogen bonds or ionic interaction. Okay, or it can also be stabilized using the hydrophobic interactions as well. Okay, in fact, there are certain research groups which have published some data which tells that it is this hydrophobic, or you can say it is this hydrophobic property of the proteins and your ligand molecule which comes into the play as the first responders of protein ligand interactions. Okay, so it is the hydrophobic interaction between the parts of the protein molecule and the ligand molecule which comes first into the play when the ligand approaches your protein. Okay, through this hydrophobic interaction, the ligand first gets entry into the active site of your protein, or wherever the binding site of the ligand is, and after it gets entry into the binding site, this ligand then fine tunes its conformation in order to form polar interactions like hydrogen bonds, or ionic interactions like salt bridges, or even coordinate interactions as such. Okay, that would further stabilize the protein ligand complex. Okay, तो कुछ लोगों का ऐसा कहना है कि this hydrophobic interaction is the first line of interaction when the ligand approaches to the protein. Okay, so this hydrophobic interaction might enable the ligand to get entry into the binding pocket of your protein, and later on it can explore some conformational freedom, and then form subsequently form the hydrogen bonds or other type of polar interactions as such in order to make your protein ligand system more stable. Okay, so in this way, this molecular hydrophobic potential, which is a term that correspond to the contribution of the hydrophobic effect in your protein ligand complex. So molecular hydrophobic potential क्या करता है? It is one of the measure, or it is one of the term that represent the hydrophobic contribution of a ligand in stabilizing the protein ligand complex. Okay, So in order to then calculate, or in order to decide ki what is the contribution of the hydrophobic interaction as such for stabilizing the protein ligand complex, you can use some free online tools like Platinum. Okay, this Platinum stand for protein ligand attraction investigation numerically. Okay, so this protein ligand interaction can be somehow be computed. Okay, and it can somehow be visualized ki what is the hydrophobic contribution of say some ligands carbon atoms for stabilizing that protein ligand complex. Okay, so this Platinum estimates the hydrophobic or hydrophilic complementarity of the ligand to the receptor binding site. Okay, and the hydrophobic contribution is mapped in the B factor column of the ligand and can be visualized in PyMol. Okay, तो ये जो कुछ भी हाइड्रोफोबिक पोटेंशियल कैलकुलेट किया जाता है वो हाइड्रोफोबिक पोटेंशियल वो हाइड्रोफोबिक कंट्रीब्यूशन कहाँ पर मैप किया जाता है? इन द बी फैक्टर कॉलम ऑफ योर प्रोटीन. डू यू रिमेंबर द बी फैक्टर कॉलम? I have told you that when I represented your protein molecule in the flat file format, I have shown you that there are 3 columns for x, y and z coordinate of each and every atom. तो उसके बाजू में दो और कॉलम थे. The one was for occupancy and the very last column was for B factor, in which B factor is usually used to demonstrate or to document the thermal vibration of atoms in the protein. So what these server or what this tool does is that it tries to map the hydrophobic contribution to the values in the B factor column. Okay, so that when you map it by B factor, you would be mapping it by molecular hydrophobic potential rather than B factor. मैं अभी आपको वो बताऊंगा कि वो चीजें कैसे होती है. Okay, so let us try to visualize. This is how you might see the molecular hydrophobic potential. Okay, so these are the two papers in which we have tried to analyze and visualize the molecular hydrophobic potential of the ligands that we have synthesized in our laboratory. Okay, so first thing and the foremost thing that you need to do is that you need to have a surface representation for your ligand. Okay, and once you are able to create the surface representation, then you need to identify ki what is the hydrophobic contribution of the atoms of that ligand molecule. Okay, then you need to provide some values to those atoms and then you can map it as shown over here. So in this color scheme, now focus on only ligand. The yellow portion of the spectrum would indicate the hydrophobic contribution, while the green portion of the spectrum would indicate the hydrophilic nature of your ligand. Okay, well now just consider this particular ligand. Okay, तो इसमें देखो whatever portion of your ligand that is exposed to the surface of the protein, that means it is it is accessible to water, therefore it cannot have hydrophobic interaction, therefore it is shown in green color, while this particular portion of your ligand is buried deep inside the protein and therefore it can have hydrophobic contribution with the protein molecule, therefore it is colored in the shade of yellow. Okay, तो ये जो spectrum है that would be in a shade of yellow, which would show highest hydrophobic contribution. On the other hand, the lowest hydrophobic contribution or towards the polarity, or you can say hydrophilicity of your molecule, would be shown in the green color, and the intermediate color would be shown in well say for example in shade of white. Okay, so now I will kind of demonstrate you how these things can be done. Okay, so before we move on to hydrophobic potential, okay, let me show this, right? So I have an output from this platinum server. Okay, so this is the protein molecule that I was talking about. Okay, now let me hide the lines representation and uh, show it by cartoon representation, so that it would look, and I would color it by rainbow spectrum, or maybe I will color it by, okay, whatever, I have colored it by rainbow. Okay, then now what I need to do, first I will show you how hydrophobicity is described. Okay, so here is the molecule. I will open it with text pad. Okay, and as you can see over here, the hydrophobic contribution has been mapped in this B factor column. This is the B factor column, right? These are the three columns that represent the XYZ coordinate. This column represent the this column represent the occupancy, while this column represent the B factor. So using this tool, Kunsa tool, this platinum tool, what we have done is that we have kind of tried to map the hydrophobic contribution, okay, in the range of 0 to 1. Okay, and whatever is the contribution of this first atom in your molecule, it would be mapped with the range of 0 to 1. So this first carbon has been provided with a value of 0 to 0.81. That means it is highly hydrophobic. Okay, if it is in a range of, well, say for example very low range, 0.98 or something, okay, then you can say ki it is low hydrophobic in nature. Okay, so in this way we map the so called hydrophobic calculation in this column. Okay, and then if we color this column by the spectrum, you can visualize the molecular hydrophobic potential that I am going to demonstrate you now. Okay, so let us see how these things are done. Okay, so now I have already loaded the protein molecule. Now I will load the ligand molecule as well. Okay, now you see that the ligand molecule has been loaded. Okay, so I will hide this lines because we want to show it in surface, and show it in surface. Okay, now it is colored by some CPK representation. Okay, maybe I will color it in a shade of white so that you can see now we have, you know, colored the entire surface by white. Okay, so now let us map the hydrophobic potential that we have calculated uh, from that platinum server, and we have mapped it as per this column. Okay, so the highest value which is present in this column would be given the green color, and the lowest value would be given the yellow color. Okay, so this is what we are going to do now. Okay, so there is a command called as this. I will explain you what this command is all about, and then I will hit enter. So you are supposed to give the command over here. So what we are supposed to do, we are using the command called as spectrum, in which we are going to use the data from B column, and we are going to color the data from B column or B factor column in a range of green, yellow, green, white and yellow. And selection all means whatever we have selected so far, it would color everything. Okay, so here is what, and if we hit enter, here it is, right? We have kind of map the hydrophobic potential as per the values,
Hmm.
right? You see, green would be the highest and yellow would be the lowest. That means it is mostly stabilized by hydrophobic interaction. दिख रहा है यहाँ पर मैक्सिमम वैल्यू किसको दी गई है, ग्रीन को दी है, राइट, एंड इट हैज बीन कलर्ड बाय ग्रीन. ओके, तो ग्रीन कलर क्या रिप्रेजेंट करता है, हाइड्रोफोबिक पोटेंशियल रिप्रेजेंट करता है. Okay, you can do the other way around as well, right? You can show it as yellow, white and green. तो ये केस में क्या हो जाएगा, yellow would represent your hydrophobic potential. Okay, it is still working. Okay, do you get this thing? सबको समझ
Yes.
में आ रहा है? Okay, in this way you can map the hydrophobic potential on the surface of protein and see कि ये देखो, it is buried deeply inside the protein, right? दिख रहा है? इसको
Yes,
कहीं
sir.
पर बाहर जाने का या फिर कहीं पर बाहर से इसको वाटर मॉलिक्यूल से कांटेक्ट होने का कोई चांस नहीं है. देयरफॉर इट इज मोस्टली स्टेबलाइज्ड बाय हाइड्रोफोबिक इंटरेक्शन. ओके,
Yes.
गुड. सो आई विल क्लोज इट. Okay, so now let us talk about this electrostatic potential now. Okay, so first let me introduce you with the concept of electrostatic potential. क्या होता है, well let पहले ये जान लेते हैं, and then we would see how can we map that electrostatic potential and what are its application in biology, right? So far the protein structure is concerned. Okay, so now there are a few things that you should know before we proceed towards discussion of this electrostatic potential. Okay, so first let us define what is electrostatics. ये तो आपने 12वीं में भी पढ़ा होगा, ठीक है. Electrostatics is a part of physics that describes interaction between two stationary charges, right? And Coulomb's law is associated with electrostatics that states that if there are two charged particles, then they might exert force on each other which is equal to the product of the two charges divided by the distance between the two charges. Okay, that is how the electrostatic force is calculated. So this electrostatic force is proportional to the product of charges on on the two particles q1 and q2, and this force is inversely proportional to the square of distance between these two particles. इसका मतलब क्या कि इलेक्ट्रोस्टैटिक फोर्स दो चार्जेस के बीच में मैक्सिमम कब रहेगा, जब उसके बीच में डिस्टेंस कम रहेगा. दो चार्जेस के बीच में अगर डिस्टेंस बढ़ता है तो वो दो चार्जेस के बीच का फोर्स जो है वो कम होता है. दिस फोर्स कैन बी अट्रैक्टिव फोर्सेस और दिस फोर्स कैन बी रिपल्सिव फोर्सेस. Okay, is it clear?
Yes, sir.
Okay, now The potential energy of such charge system, what we call it as simply energy or potential energy, that is created by electrostatic interaction between any two charged particle is equal to the multiplication between the two charges divided by the distance between the two charges. Okay. So now let us there is this potential energy of the charge system. This there can be a charge system whose force can be defined by the distance as well as the charges that are present in your system. So now it is time for you to get familiarized with the concept of electrostatic potential. Okay, so the electrostatic potential is the quantity that is related to the potential energy of a charge system. अब वो कैसे कि you can define the electrostatic potential as you can say that this electrostatic potential is created by a system of charges at a particular point in space with the coordinate, we'll say x, y, and z, and this potential is equal to the change in the potential energy that occurs when one plus one ion is introduced at this point. Okay, so this is how we define. I will illustrate you how things would work over here. So say for example, we have a charge system as you can see over here. Okay, consider a molecule which consists of electron density which is shown in cloud and several positively charged nuclei that are present in this electron density cloud as such. Okay, so similar would be the situation of protein molecule as well. बरोबर है. Protein molecule क्या है at 3-dimensional state? It is nothing but some positively charged nuclei which are surrounded by the electron densities of those positively charged nuclei. बरोबर है. ओके, सो इस तरीके से अपन अपना चार्ज सिस्टम फिलहाल डिफाइन कर रहे हैं विच वुड बी एनालॉगस टू आवर प्रोटीन सिस्टम एज वेल. Okay, so now suppose we want to know the electrostatic potential of this molecule. Okay, of this molecule create this molecule, and if you want to know ki what electrostatic potential this molecule can exert at this particular point which is having the coordinate of x, y, and z. ठीक है. So we can obtain the potential by introducing one plus charge at this particular point. Okay, अपना objective क्या है कि we'll say we can compute what is the electrostatic potential that is exerted by this system at this particular point. अगर ये आपका ऑब्जेक्टिव होगा तो आपको क्या करना पड़ेगा, ये वाले पॉइंट के ऊपर में आपको एक +1 चार्ज इंट्रोड्यूस करना पड़ेगा. ओके, और ये पॉइंट का आपको कोऑर्डिनेट पता रहना जरूरी है, उसका लोकेशन पता रहना जरूरी है विद रिस्पेक्ट टू योर चार्ज सिस्टम. Okay, so the new picture would look like. So अपना objective क्या है अभी? We want to calculate the electrostatic potential that is exerted by this particular system at this particular point. In order to do that, we need to place a +1 charge at this particular point. Okay, so now how can we calculate the electrostatic potential? तो सबसे पहले एक point के बारे में बात करते हैं. ठीक है, we will first go on talking about this point as such. Okay, so whatever the change in free energy that happens by introducing one charge at this particular point around our charge system. Okay, so the change in the energy is simply the potential energy that is created by interaction of this by interaction of charges that are present in our system with the +1 charge that you have just introduced. Okay, so we can calculate this energy by formula जो यहाँ पर दिख रहा है. ठीक है, how would we read this equation now? It is the change in energy when a +1 charge is introduced with or introduced along the Q molecule divided by the distance between the plus 1 charge and the Q molecule. This is what it means. R is the distance of this Q molecule that you have already placed over here, which is having plus 1 charge, right? And this is the Q molecule, which is our interest of, you can say, system. Okay, तो प्लस 1 चार्ज यहाँ पर है, Q मॉलिक्यूल यहाँ पर है, प्लस 1 चार्ज का well, you can say what is the distance of this plus 1 charge from your Q molecule. That would give you an idea of the change in free energy that is imparted by placing this plus 1 charge around this Q molecule at the distance r. Okay, so now this formula calculate the energy for one charge in the molecule Q molecule, right? तो ये क्या करता है? It calculates the इलेक्ट्रोस्टैटिक पोटेंशियल फॉर वन पर्टिकुलर चार्ज. तो आपके प्रोटीन में ढेर सारे चार्ज रहने वाले हैं. ओके, सो यू नीड टू अकाउंट ऑल ऑफ द चार्जेस दैट इट माइट हैव कंट्रीब्यूटेड, ओके, टुवर्ड्स द फ्री एनर्जी. तो इसके लिए आप क्या करते हैं, वो सारे के सारे चार्जेस का सम कर देते हैं. इफ यू हैव n charges in your Q system, molecule Q system, then what you do, you sum up all the electrostatic potential terms that you have calculated as you have calculated for a single charge. Okay, and ये summation क्या बताते हैं, you are going to do the same electrostatic energy calculation right beginning from molecule 1 till there are n molecule in your n charges in your Q molecule, and the resulting change in energy is called as electrostatic potential. Okay, so this is how you can visualize it. Okay, so now this is a kind of global phenomenon, right? So the electrostatic potential which is exerted by introducing this plus charge along this much area of your protein might be different than the electrostatic potential that is exerted on this charge, on this charge, this charge or this charge. हर एक का electrostatic potential अलग रहेगा, right? बरोबर है?
Yes, sir.
So
Yes.
the display of electrostatic potential is a difficult problem, क्योंकि हर एक पोजीशन का electrostatic potential अलग रहेगा. Okay, we cannot look at the potential everywhere simultaneously, right? क्योंकि हर एक जगह पर अलग रहेगा. So we must
Yes,
select
sir.
some point of interest first and display the potential at these points. Well, say for example मैंने simple water molecule अगर consider किया है, okay, and I mapped this water molecule with a surface, तो मुझे पूरे water molecule के surface पर electrostatic potential अगर बताना होगा तो it is a difficult task, right? So what I do is that I select some point. Okay, I would select this point on the surface, this point on the surface and this point on the surface. Okay, so what we do is that in order to visualize the electrostatic potential data, we select the points located on the molecule surface and calculate the potential at these points. Okay, and then the software assigns color to this point according to their potential. तो सबसे पहले आपका सॉफ्टवेयर क्या करेगा, इट वुड ट्राई टू फिगर आउट कि आपका जो इलेक्ट्रोस्टैटिक पोटेंशियल आपके चार्ज सिस्टम पर है, सबसे ज्यादा कौन से पॉइंट पर है और सबसे कम कौन से पॉइंट पर है. ओके, व्हाट आर द पॉइंट्स इन योर सिस्टम on the surface system where there is maximum electrostatic potential, and what are the points in your system where there are lowest electrostatic potential. Okay, so in this way the electrostatic potential points are decided, and these points are then colored as per the visible spectrum. Okay, so it is possible, अपना visible spectrum में क्या होता है, V I B G Y O R, VIBGYOR, right? So it is possible that one might color the points with highest electrostatic potential in the shade of violet, and the point which is having the lowest electrostatic potential in the shade of R. तो बीच में जो कोई भी इंटरमीडिएट वैल्यूज रहेंगी उसको अपन I, B, G, Y, O के रेंज में कलर कर सकते हैं. दैट इज व्हाट इज शोन इन ओवर हियर, राइट. तो वाटर मॉलिक्यूल में सबसे ज्यादा इलेक्ट्रोस्टैटिक पोटेंशियल, well say for example a negative value might be present over here and positive value might be presented over here, and whatever the intermediate values जो आजू बाजू का सरफेस के ऊपर है उसको विबग्योर के हिसाब से यहाँ पर कलर किए वाला है. ओके, so in such a way the resulting rainbow colored surface is called as potential map. इसे क्या कहते हैं अपन? We call it as potential map. Okay, so now how softwares create such electrostatic potential map? Okay, so first the program surveys all the potential on the map and finds the most negative and most positive potential. These region are colored, well say for example red and dark blue respectively. Okay, and the intermediate potential are then assigned according to the color spectrum that you are interested in. Okay, so the red would be most negative, followed by yellow, green, light blue, dark blue. Dark blue would be the most positive portion of your protein. Okay, so now what might be the factors that affect the electrostatic potential? Okay, now अभी ये डिस्कशन से आपको एक चीज तो पता चली गई होगी कि इलेक्ट्रोस्टैटिक पोटेंशियल काय के ऊपर में डिपेंड करेगा. इट वुड डिपेंड ऑन वेयर एग्जैक्टली दैट चार्ज इज प्लेस्ड around your protein system or around your charge system. Okay, so it would say that the electrostatic potential would depend on the charges exist in the molecule and how they are distributed. कि कौन से चार्जेस आपके मॉलिक्यूल में है. आई एम टॉकिंग अबाउट द प्रोटीन सिस्टम नाउ. तो प्रोटीन सिस्टम का इलेक्ट्रोस्टैटिक पोटेंशियल कैसे वेरी होगा? दैट वुड डिपेंड ऑन हाउ चार्जेस आर डिस्ट्रीब्यूटेड अलोंग इट्स सरफेस. ओके, और वो कैसे है, whether they are deeply buried in the protein surface or these charges are exposed to the surface protein, right? So where these charges exist and how are they distributed. It also depends on where exactly you place that xyz point that you calculate. Now if we select the point where +1 charge is attracted by the molecule, the potential over there would be negative, right? On the other hand, if we select the point where the +1 charge is repelled, the potential over there would be positive. बरोबर है?
Yes, sir.
तो देखो +1 चार्ज अगर अट्रैक्ट हो रहा है इसका मतलब क्या है कि आपके सरफेस के ऊपर नेगेटिव चार्ज है, राइट?
Yes, sir.
Therefore this +1 charge would be attracted and therefore we would color that surface as negative, right? And if this +1 charge after placing it on the surface of protein, if it is repelled, इसका मतलब क्या? +1 charge कब repel होगा? जब सरफेस के ऊपर भी +1 charge रहेगा,
Yes, sir.
right? And therefore the surface of the protein would be colored as positive. Okay, and it is the magnitude of this positive and negative potential that would eventually decide this color. Okay.
Yes, sir.
Okay, तो ये सारी झंझट अपन प्रोटीन मॉडलिंग में क्यों कर रहे हैं? ये ठीक है, ये फिजिक्स के हिसाब से चीजें ठीक है, बरोबर है. तो इसका प्रोटीन मॉडलिंग में क्या एप्लीकेशन रह सकता है, राइट? सो द थिंग इज दैट दिस इलेक्ट्रोस्टैटिक पोटेंशियल ऑफ अ प्रोटीन मॉलिक्यूल इज अ लॉन्ग रेंज फोर्स, राइट? ये क्या है? लॉन्ग रेंज फोर्स है, ठीक है. एंड दिस लॉन्ग रेंज फोर्स आर द वंस दैट इन्फ्लुएंस द बिहेवियर ऑफ अदर मॉलिक्यूल इन द एनवायरनमेंट, बरोबर है. तो ये इलेक्ट्रोस्टैटिक पोटेंशियल, ये जो लॉन्ग रेंज फोर्स है, ये क्या डिसाइड करेगा कि आपके प्रोटीन के आजू बाजू में कौन भटकेगा कौन नहीं भटकेगा, राइट? What kind of chemical species would your protein molecule allowed to come closer to it, or what kind of chemical species will not be allowed to come closer to it. And this electrostatic potential has its range of around 15 Armstrong. तो इलेक्ट्रोस्टैटिक पोटेंशियल आपके प्रोटीन का कहाँ तक डिफाइन कर सकता है? उसके 15 एमस्ट्रॉन्ग आजू बाजू तक. कि उसके 15 एंगस्ट्रॉम आजू बाजू में कौन सा केमिकल स्पीशीज आ सकता है और कौन सा केमिकल स्पीशीज नहीं आ सकता, राइट. सो इट गिव्स योर प्रोटीन an ability to decide कि okay is this molecule my substrate, हाँ तो उसको अंदर आने दो. Right.
Yes.
So in this way protein molecule itself can identify what chemical species would be allowed to come closer or what chemical species would be repelled away. Okay. So the nearby charge group with the protein may cause the pKa value, ये तो आपको पता ही है, the pH at which an acidic or basic group loses or gains the proton, right. So this pK value of any amino acid shift that would create a chemistry necessary for the molecule to perform its chemical function. बरोबर है. So
Yes, sir.
जैसे ही well say for example at physiological condition if an aspartate if an aspartate loses its if an aspartic acid loses its proton and it gets a negative charge, right. तो वो पर्टिकुलर पॉइंट पर वहां पर एक इलेक्ट्रो निगेटिव और इलेक्ट्रोस्टैटिक निगेटिव इलेक्ट्रोस्टैटिक पोटेंशियल डेवलप होगा and this negative electrostatic potential might attract the positively charged group जो उसका substrate या inhibitor रह सकता है, right. बरोबर है.
Yes, sir.
Yes.
This is the
Yes.
application, right. So computing the electrostatic potential of protein structure allows you to predict quantities such as individual amino acid pK values. You can calculate the solvation energies and approximation of intermolecular binding energies. ओके तो ये सब चीजें कैलकुलेट करने में इलेक्ट्रोस्टैटिक पोटेंशियल का बहुत बड़ा रोल होता है, right. Well say for example if your protein is a DNA binding protein तो उसका DNA बाइंडिंग रीजन में कैसा इलेक्ट्रोस्टैटिक पोटेंशियल रहना चाहिए.
Positive.
पॉजिटिव रहना चाहिए, राइट. व्हाई बिकॉज DNA हैज नेगेटिव चार्ज,
Negative charge.
नहीं क्या, ओविंग टू इट्स फॉस्फेट ग्रुप, right.
Yes.
So if the surface of your protein is having negative electrostatic positive electrostatic potential then it might attract the DNA molecule or it well so for example it can recruit itself toward the DNA molecule, right.
Yes, sir. Yes, sir.
Okay, so this is how the electrostatic potential of a protein might look like. Okay, बात समझ में आ रही है आपको. तो सॉफ्टवेयर कौन से
Yes.
होते हैं ऐसे इलेक्ट्रोस्टैटिक पोटेंशियल जनरेट करने के लिए. Okay, there are softwares like grasp, okay, and there are softwares like abps. ये जो फिगर आपको दिख रहा है यहाँ पर, well that is calculated using pymol. उसमें abps का plugin आप इंस्टॉल कर सकते हैं. Okay, there is also another software called as delphi that can calculate such electrostatic potential. तो निगेटिव इलेक्ट्रोस्टैटिक पोटेंशियल यहाँ पर रेड कलर से बताया गया है और पॉजिटिव इलेक्ट्रोस्टैटिक पोटेंशियल यहाँ पर ब्लू कलर से बताया गया है, राइट. सो दिस इज व्हाट आई वांटेड टू से, राइट. So maybe I will show you an example where I have implemented this electrostatic potential in my own research. Okay, so I will show you one of my paper maybe जिसमें I remember मैंने किया था कुछ ऐसा. It was in this molecular diversity paper. Just a minute, I am kind of exceeding my time, but I will just show you कि how I have used this electrostatic potential in my own research. Okay, इससे आपको भी अंदाजा आएगा कि आप ये चीज़ कहाँ पर कर सकते हैं. Well, this particular paper we have published last year 2020 में है and it is all about the structure of this threonine synthase from Leishmania major. Okay, and we have proposed the structural model of this paper of this protein threonine synthase, and somewhere over there in while discussing the catalytic activity of this protein, देखो यहाँ पर, okay, so now it is a PLP binding protein, right? तो PLP के ऊपर में कौन सा चार्ज रहना चाहिए? इसमें फॉस्फेट ग्रुप है तो इसमें निगेटिव चार्ज रहना चाहिए, राइट? और इलेक्ट्रोस्टैटिक पोटेंशियल यहाँ पर क्या है? वी हैव शोन द पॉजिटिव इलेक्ट्रोस्टैटिक पोटेंशियल बाय ब्लू, राइट? तो यहाँ पर जो कुछ भी अमाइनो एसिड रेसिड्यू है जो PLP यहाँ पर आकर अटैच होने वाला है, it is lined by all positively charged amino acid like arginine, lysine, histidine. Okay, so this particular region where this PLP goes and attaches, जो यहाँ पर बताया गया है, well it is surrounded by positive amino acid residues, ठीक है, and therefore the electrostatic potential over here is blue in color. Okay, तो यहाँ पर electrostatic potential blue है, इसका मतलब क्या है? This particular region can attract the PLP or the substrate or the prosthetic group towards itself. It will hold it fast so that this PLP can remain over here, so that it can form its covalent bond with this lysine residue इसके साथ में. Okay, तो ये लाइसिन के साथ में इसका कोवेलेंट बॉन्ड तैयार होने के पहले पहले उसको पकड़ के तो रखना पड़ेगा, then and then it can form कोवेलेंट बॉन्ड, राइट? तो पकड़ के रखने का काम कौन करता है? दिस इलेक्ट्रोस्टैटिक पोटेंशियल करता है. Okay, so this is one of the application. You can read this paper for more details of it, right? So we have done a lot of things. Good. So this is all for you today. Okay, now I am stopping this recording. Okay.